Hello students, welcome to EPG Pathsala. I am Dr. Hari Mohan from Center for Medical Biotechnology, Maharshi Dhyan University, Rohtak, Haryana. And today we will discuss about the module Sperm and Embryo Sexing and Disease Transmission under the paper Animal Cell Biotechnology. Learning objective of this module is to ascertain the significance of sperm and embryo sexing. What are the methods which can be used for sperm sexing? What are the latest development in embryo sexing? And what kind of disease can be transmitted by sperm and embryo sexing? Predetermination or pre-selection of sex is a priority since centuries. Human being is looking for the technologies which can be used for determining the sex of embryo before its implantation, so that we can change the sex ratio. If we say in case of dairy industry or in case of meat industry, it is always desirable to have a sex of specific kind. For example, if we take example of dairy industry, the farmer is always willing that a female calf should be born, so that the milk production may increase and the number of herd animals may increase. So in case of dairy industry, excess of male are considered as waste because they will consume the resources which are limited. On the other hand, in case of meat industry, male are preferred. Male calves are preferred or female calf because males have better food conversion efficiency and a higher growth rate. In case of dairy meat industry, female calf, they are not desirable. So if it become possible for human being to pre-select the sex, that will definitely increase the efficiency as well as profitability of animal husbandry. But there are certain issues, there are certain ethical issues which are associated with predetermination of sex. So that also need to be taken care of. So predetermination of sex can be carried out by two main methods. They are sperm sexing and embryo sexing. Sperm sexing means sorting of sperms on the basis of the X or Y chromosome. So we can say X kind of sperm and Y kind of sperm. So question you can ask is, what is the difference between X sperm and Y sperm? It has been reported that there are some kind of differences between X and Y sperms, and main difference is on the basis of DNA content. It has been found that the X sperm contains slightly more amount of DNA as compared to Y sperm. So on the basis of DNA content, they can be differentiated. Then on the basis of concentration of X encoded genes, because in female the X genes will be expressed in more concentration. So if we can estimate the X encoded genes, that can also serve the basis of sperm sexing. Then presence of Y chromosome specific gene. If we are able to detect genes which are specifically present on Y chromosome, that can also be used for sperm sexing. Apart from these methods which differ sperms on the basis of their size, on the basis of their morphology, are also used. So the different morphology methods are also used for sexing of sperm into two different types. Sperm sexing can be done by various methods. Important methods are albumin gradient, Percol gradient, swim up procedures, electrophoretic separation, and flow cytometry. These methods are based on a basic principle that X sperm and Y sperm are somehow different. For example, Percol gradient and albumin gradient is based on the difference in the size and mass of X and Y sperm. Y sperm is slightly smaller than X sperm, so difference in motility can be used for separation of X and Y sperm, although that can be very difficult. Swim up procedure is also based on the swimming capability differences between X and Y sperm. Electrophoretic separation is based on the difference between the charges on X and Y sperm. The technique which is very important for sperm Sexing is flow cytometry, and this is the latest technique which is used commonly for sorting of sperms. X and Y sperm differ in DNA content. This slide indicates the difference for Jersey cattle is 4.22, means the X sperm contains more DNA content as compared to Y sperm, and this difference in DNA content can be utilized as a principle for separation of X and Y sperm. Similarly, for Brahman breed, it is 3.7%. The figure number 2 indicate the X and Y sperm DNA content difference among the mammalian species. So it indicates that in all the animal species and human being also, there is a difference ranging from 7.5% to 2.3%. So maximum difference, that is 7.5%, is chinchilla, and the minimum difference is in possum. So this picture indicates the principle how Perkol gradient and swim procedure work. In Perkol gradient, the initial sample of unsorted spermatozoa, they are laid over a different Perkol concentration, that is 45% Perkol and 90% Perkol. When this mixture is centrifuged at 700 G for 30 minutes at 25 degrees Celsius, it results in accumulation of viable spermatozoa at the bottom of tube, whereas the non-viable and abnormal spermatozoa will accumulate at interface. So this viable spermatozoa can be isolated, and then X and Y sperms can be separated on the basis of their difference in swimming capability. The second method, that is swim up procedure. In swim up procedure, the sperm wash media is used and initial sample is deposited in the bottom of tube, and it is allowed to stand for 30 to 60 minutes at 37 degrees Celsius. So the sperms which are motile, they will try to move upward, and viable spermatozoa can be collected. So difference in motility form the basis of separation of X and Y spermatozoa. The main disadvantage with this traditional technique, whether it's Procol gradient, whether it's swim up procedure, is the many part of sperm motility is lost, because sperm will expand its energy in moving up or down. So the capability of sperm to fertilize ova decreases. So almost we can say X sperm contains 4% more DNA as compared to Y sperm. Across various species difference, the difference may be as low as 3 or as high as 4.5% in different cattle species. So albumin gradient, Perkol gradient, and other traditional method can be used for separation of X and Y sperm. Y sperm is slightly smaller in size and has greater downward swimming velocity. So this principle can be used for separation of X and Y sperm. Sperm at the bottom of column are rich in Y sperm. So if we collect sperms from bottom of tube, they will be rich in Y sperm. So FACS is commonly used for sorting of sperm. Today, if we say, if we want to separate X sperm and Y sperm, fluorescent activated cell sorter technique is used very commonly. So this technique is based on the principle that the DNA content of X sperm and Y sperm is different. The X sperm contains more DNA as compared to Y sperm. So when these sperms are stained with fluorochrome, for example, acridine orange, methamycin, OIST 33258, or DAPI, so these stains will stain the nucleus, the chromatin material. So if DNA content is more, like in X sperm, so that will be stained more intensely as compared to Y sperm. So accurate identification was first reported by Garner et al. In 1983 by using FACS technique. So the technique utilized staining of sperm with certain dyes which stain their genetic material, and then they are passed through a beam of laser. The signal at 90° is used to determine the sperm orientation, whereas the signal at 0° is used to determine the DNA content. So depending on the DNA content, whether DNA content is more or it is less, different charges are given to this sperm, and on the basis of charge, they can be separated in two tubes. Means X sperm and Y sperm can be sorted. In some cases, it may be impossible to determine that whether the sperm is X or Y, so that will go in the third tube. That is unpredictable. Embryo can be sexed by two methods. One is invasive and second is non-invasive. Invasive methods include injecting a needle in the embryo and taking out certain cells. So this procedure may be dangerous. It may kill embryo or it may lead to abnormal development. Important invasive methods are stereotyping, bar body detection, FISH for Y chromosome specific sequences, and PCR based method. Important non-invasive methods are detection of HY antigen, which is specific for male embryo, concentration of X-linked enzymes, means expression of genes which are present on X chromosome only, and differential growth of male and female embryo. Although theoretically it is possible that male and female embryo grow at a different rate, but it becomes very difficult to differentiate that whether the embryo is male or female only on the basis of their growth. So after synchronization and superovulation, oocytes are collected from the superior quality female. The number of oocytes, they are fertilized in vitro in the lab by providing almost the same environment inside the body. So after in vitro fertilization, embryos will be formed, and these embryo may be of different cell number. So most commonly morula is used for sex determination, but morula as well as blastocyst can be used for sex determination. After blastocyst, embryos are not used for sexing. So this picture indicates that using a injection, certain cells of embryo like inner cell mass or trophobacterium can be taken out, and these few cells, they can then be cultured or they can be used for karyotyping, or DNA can be extracted from these cells and can be used for setting up PCR reaction. So this is the disadvantage associated with invasive techniques, as it may lead to damage to the embryo and that may result in death of embryo. Karyotyping of the cells extracted from embryo serves as a traditional method of determining the sex of embryo. So karyotyping is done at metaphase stage. Cells which are isolated from embryo, they are lysed osmotically, and preparation is done after fixing and the slide is stained. So on the basis of difference in X and Y chromosome, the bending pattern can be observed, and it can be ascertained whether the embryo is male or female. Advantage with this technique is it is a inexpensive technique, easy to perform, it requires no sophisticated equipment, and it can identify chromosomal abnormality also. Accuracy of karyotyping method is high, and reagents are inexpensive and easy to obtain. But it also suffers with certain disadvantages, as it is a invasive method and it can lead to accidental harm to embryo that can reduce the viability and conception rate of embryo. Poor spreading and overscattering of metaphase chromosome also pose a hindrance in detection of sex by using karyotyping. Identification of bar body also serves as an important marker of sex. Bar body is a heterochromatin. In case of female, one chromatin become inactive and it become a dark stained body adjacent to nuclear membrane. Bar body will only be present in female embryo. It was Edward and Gardner in 1968 who first time demonstrated sex by bar body in the trophoblastic cell in rabbit. So on the basis of presence or absence of bar body, sexing can be done. Advantage of this method is it is simple and rapid technique. Disadvantage, the granular nature of cytoplasm prevent the detection of bar body. All cells may not have bar bodies. So presence of bar body depend on stage of cell and the direction of cell during fixing. So it cannot be a confirmatory method for sex of embryo. In addition, it is also not possible to determine the sex in cattle, sheep, goat and pig because their cytoplasm contains granules and it requires large number of cells. So that can also damage the embryo. Fifth technique, that is fluorescent in situ hybridization, is a important technique for determining sex of embryo. Y-specific probe can be used for detection of Y chromosome in the given cell. This technique utilizes fluorescent labeled probe that will bind specifically to the complementary gene. After proteinase digestion of chromosome that results in removal of proteins, histone proteins, the specific probe labeled with fluorescent dye is allowed to hybridize with the genetic material. If Y chromosome is present, fluorescence can be observed. If Y chromosome is absent, in case of female, there will be no fluorescence. So sexing of bovine embryo can be done in 30 hours. The beauty of this technique is it can be done with very little DNA. So it requires very less number of cells for determining the sex. Another advantage is it is highly accurate and a very high percentage of embryo can be sexed by using this technique. Disadvantage associated with this technique is it is quite complicated, expensive and time consuming. It is also limited by its accessibility of the biopsy of embryo. Sexing of embryo can be done very efficiently with polymerase chain reaction. Polymerase chain reaction is based on three basic steps that are denaturation, annealing and elongation. This technique can amplify even a single copy of DNA into millions of copies in a time period of 2 to 3 hours. So it is based on the principle of selective primers, means which are primers which are specific for a specific sex. So we can target X or Y, but it is more important to target Y because Y directly means male. So a portion of Y chromosome, or in other words we can say genes which are specific to Y chromosome, can be targeted. Primers against these genes can be designed and these genes can be amplified using PCR reaction. The technique for PCR reaction has been standardized for cattle, sheep, goat and pig. In case of goat, it is gene amylogenin which has been standardized, and this is a highly reliable test for determining sex in goat. This technique have several advantages over the other techniques. It is a sensitive, accurate, highly reliable technique and it produces results with high efficiency. Apart from testing the sex of embryo, this technique can also be used for determining any kind of genetic abnormality, whether there is some kind of differences in the pairing of chromosomes or whether there is some kind of deletion or translocation. So this technique can be used for screening of embryo for any kind of genetic disease. This technique can be used for screening higher number of embryo in short time. So as compared to other techniques, it is a rapid technique and we can screen a larger number of embryos in shorter duration. Important about this technique is even from a single cell, the DNA extracted from a single cell can be used for detecting whether it is a male embryo or it is a female embryo. In spite of all these advantages, This technique also suffers with certain disadvantages as it requires technical skill. So only a technical person which is expert in PCR can do polymerase chain reaction, and it is important if during handling of sample certain kind of contamination is there from the environment, so there are chances of false positive result. So even a single cell from outside can change the results, and this technique require accessibility of embryonic material, means it is a invasive technique. Apart from that, it also require a machine, that is PCR machine, that is also costly. So it also suffers with certain kind of disadvantages, but the advantages outweigh the disadvantages. There are more advantages as compared to the disadvantages. Non-invasive methods, which don't involve puncturing the embryo and taking out certain cells. An important non-invasive method is detection of X-linked enzymes. As in case of female, two X chromosomes are present, and the genes which are present on X chromosome will be expressed more in female, although some of the DNA become heterochromatin as bar body. So the genes present on X chromosome, for example, glucose 6 phosphate dehydrogenase, hypoxanthine guanine, phosphoribosyl transferase, phosphoglycerate kinase, alpha galactosidase. So these enzymes will be expressed in high concentration in the female embryo as compared to male embryo. So high concentration indicate female, whereas low concentration will indicate male. Advantage of this technique is high number of embryo can be sexed in shorter time. But disadvantage is it may take long time to estimate enzyme, and for that time we have to keep the embryo outside the body. So it may cause a damage or it may cause toxicity to the embryo. Detection of HY antigen. HY antigen is a male specific factor, and it can be detected by using specific antibody. The embryos are incubated for 30 to 60 minutes with antibodies, and then an additional 30 to 60 minutes with first antibody containing a fluorescent dye. Embryos are then briefly examined with a fluorescent microscope. If it is a male embryo, fluorescence will be observed. In female embryo, no fluorescence will be observed. It is important to note that this HY antigen is expressed only in the early stages. So at 8 cell stage, at modular stage, it is readily detectable, but as the embryo grows and it becomes blastocyst, its detection become difficult. So if we are using HY antigen method of embryo sexing, it is always advisable that early stage embryo, that up to modular stage, it should be used. Advantage of this method is it is simple to perform and rapid enough to allow field use. Although the accuracy with cattle embryo is at best 80 to 90%, speed and lack of need of biopsy embryo is the beauty of test. So we don't require biopsy and it is quite rapid. But it also suffers with the disadvantage that we require a fluorescent microscope. So sexing technology have many applications and which can change the face of animal husbandry. By sexing techniques, we can change the male to female ratio in farm animal. So according to the need of farmer, we can have more number of females or more number of males, whether it is a dairy industry or meat industry. In this way, the output of milk and meat can be increased. It will also decrease the cases of free martinism. So free martinism is a process in which the female becomes sterile due to some genetic reasons. So these genetic screening can be performed early before implantation of embryo. These techniques can also be used for preparing quality bulls, and these quality bulls can be used for progeny testing and mass improvement of non-descript breeds. So this technique will prove vital in upgrading of non-descript breeds. In addition, this technique can be used for preservation of rare breed of farm animals. So the breeds which are less in number and which have high productivity can be preserved for future. The endangered wild species which is feared to get extincted in near future can be preserved by this method, and the species which have already extincted from earth, these techniques have potential to take back those animals on the earth again. So now scientists are trying to have dinosaurs back. If we are able to get their DNA intact, it may be possible to have these species back on earth. So question one can ask is whether this sexing technology is really required in Indian scenario. The answer is yes, because more than 40% of Indian population is dependent on agriculture. So we can say India is a agricultural economy. It contributes more than 11% to GDP. So these most of the Indian population which is dependent on agriculture and animal husbandry, so they require some kind of new technology. Due to decrease in land for fodder and animal rearing, there is a greater challenge in front of us to increase the productivity of animal. So with the less land, we have to increase the productivity, because excessive male, they are consuming the fodder. So we need to decrease the number of unproductive animals. In case of dairy industry, it is male which is not required. So by using this technique, we can eliminate the unnecessary males which are consuming the limited resources. And this is the way that only females calf will be born, and these can be used for increasing milk production for the country population. Similarly, for meat industry, we can target the male calves because they have higher feed conversion efficiency and will gain body weight rapidly. So in the present scenario, this kind of technology is a need of hour for Indian population. So although these sexing techniques have many advantages, they also suffers with certain shortcomings. They also suffers with certain kind of pitfalls. And important shortcomings are lack of proper infrastructure facilities under field condition. So these techniques seems quite sophisticated. So whether they can be performed under field condition, it is still questionable. Right now, their cost is also very high. So most commonly used technique facts result in very costly X sperm or Y sperm. Low level of training and education also makes this technique inaccessible right now. And there is large gap between research and field. In laboratory, scientists claim big, but on field, they actually don't yield the required results. And there is lack of indigenous tools and techniques. So every country, according to their need, have to develop the indigenous tools and techniques so that it become cheaper and it become practical to apply these techniques in a field condition. So these techniques seems attractive, but these techniques also suffers with some kind of disadvantage. An important disadvantage is risk of disease transmission. So while doing sexing of sperm or sexing of embryo, it is possible that contamination of certain kind of pathogen may occur. So important pathogens which can cause contamination of sperm or embryo is infectious bovine rhinotrichitis, infectious pustular vulvovaginitis, bovine viral diarrhea, brucellosis, leptospirosis, trichomoniasis, foot and mouth disease, mycoplasmosis, swine erysipelas disease, hog cholera. So these infectious agent may contaminate, and it is possible that if the bull is suffering with any of these disease may spread very rapidly, because semen of a single high quality bull will be used for thousands and thousands of female. So if the bull is suffering with certain kind of disease which could not be detected, that will lead to massive spread of disease across a larger population. So we need to be very careful that the bull we are taking for a source of semen or semen donor should be free from all type of disease. So if we talk about the current status of sexing techniques, embryo sexing has been standardized for cattle, sheep, pig, horses, goat, buffalo, and humped cattle. Although differential growth of male and female embryo has been detected, but it cannot be applied for sexing of embryos. We have standardized the sexing technique to the extent that even a single cell can be used for determining sex of embryo. Rapid sexing within 2 hours can be done by using multiplex PCR, and these multiplex PCR use BOV 97M bovine 1.715 satellite. So many kind of satellite markers can be used for determining sex. Non-electrophoretic method of PCR sexing has reduced the time requirement to less than 2 hours. So very rapidly by using PCR technique we can determine the sex of embryo. With advancement in techniques, newer techniques like nested PCR and loop mediated isothermal amplification seems promising to take the technique to field condition. It has been found that hyperglycemia in the embryo or sperm induced apoptosis. So we can summarize this module as there are many techniques which can be used for sexing of sperm and sexing of embryo. But these techniques require excellent and careful animal management. These techniques are not self-sufficient in itself. Apart from these techniques, we have to manage the feed, we have to keep the animal disease free, and we have to follow very good animal husbandry practices. As it has been reported that these sperms which are sexed with the sperms of particular sex, they have low fertility power as compared to the normal sperm, as during the procedure of separation some kind of deformity may occur. So it has been reported that their fertilization power is less as compared to the normal sperms. So for that reason, the animal husbandry practices should be very good, and the animals should get conceived even with the smaller number of sperms. The timing of artificial insemination is very important in this case, as this technique is very costly, and the sperms which are sexed, they are very costly as compared to normal sperms. So the technique should be very precise, and artificial insemination should be done timely. Now if we say the technique is very costly, but if the cost decreases during course of time, the applicability and acceptability of this technique will increase, and definitely when the technique will be used in field condition, it will directly increase the profitability of the farmer. Apart from these techniques, effect of difference in genotype of the female animal, vigor of sire or semen, heat tolerance should also be considered for tropical and subtropical region, as the techniques have been standardized for temperate region. So this technique need to be standardized for the hot areas or subtropical region. This sex semen could markedly increase the genetic improvement rate. So in a very short span of time, we can improve the genetic constitution of a animal population in a given area. Though these techniques have a number of advantages, and these techniques seems promising. If we can change the constitution of animal, so we can order as per our requirement. If we require female animal, so we can have female animal in our herd, or if we require male animal for meat production, we can have more number of male animals. So it seems very good, it seems very mesmerizing, but we need to be very careful that these techniques also carry a risk of disease transmission, as during processing of sperms or ova there are always chances of contamination. And if that contamination is with some kind of bacteria, for example brucella, or some kind of virus such as BVD virus, so that can also be a source of infection to many females. So we have to be very careful that there should be minimum chances of contamination so that the technique can fulfill its promises. In future we may see the effect of this technique, as it will change the way animal husbandry practices are followed. Thank you very much.